Today we're going to talk about eyes. I love talking about the eye. It is truly a remarkable organ. Behold such a sight. Angels sing in their design, eye and ear sublime. Yeah, the next two lectures we get the chance to talk about two of the most remarkable aspects of being human, our ability to visualize something. What is it that is happening that we are experiencing when we see? What is it that's happening when we experience a sound? Where is that being internally represented? What are the mechanisms by which we are translating these light images and sound waves into experiences? So a little of the history. Before this point, I have spoken mostly about Eurocentric medicine, and that weighs upon me. That is not because of my lack of interest in or respect for medicine from other places in the world, other points in history. It's just that the allopathic medicine that we talk about in Western schools is heavily influenced by the European tradition. But this is a lovely opportunity for me to blow your minds with this guy, Sushruta. He was an Ayurvedic scholar who lived in Kashi almost 3,000 years ago. So incredibly, this guy had described at the time in these Ayurvedic texts 76 distinct ocular diseases. That's pretty impressive. Of those 76 different ocular diseases, he was able to surgically treat 51 of them 3,000 years ago. He removed cataracts almost 3,000 years ago. Here are some depictions, some drawings of some of the earliest ocular surgery tools. So, while 3,000 years ago, while Europe was still utterly in the dark, this man here was truly ahead of his time, an enlightened individual. So, notable person. It took the Europeans until this guy, John Freak, who wins points for interesting hairdo. It took Western medicine until this gentleman to catch up to Sushruta. He represented the advent of modern ophthalmology, and it was with the establishment of this place called Mooresfield. Anybody who's interested in ophthalmology, that would be a fun place to visit if you're ever in London. So this was where ocular disease and treatment was first described in Europe. All right. We're going to have to go through just a little bit of anatomy so that we can talk about the eye, because I'm going to talk about sort of macroscopic physiology and then a little bit of microscopic molecular physiology here today. But we're going to just talk about the basic optical functioning of the eye. To do that, we have to get the anatomy down. So looking at the eye, the first thing to recognize is that there are three layers. We see this outer layer. We'll talk about them in detail in upcoming slides, which is called the sclera. Then there is the uvea, which is this middle layer depicted in orange. That is a vascular layer. This outer one is a fibrous layer. And then there's the neural layer, the retina on the inside, the deepest part of the eyeball. We have this sclera blending into the cornea. It's a clear fibrous layer on the outside of the eye. Light passes through it, goes through this anterior chamber of the anterior cavity. There are two cavities in the eye, and one of them has two chambers. There's the anterior cavity, which is up here, and then there's the posterior cavity, which is back here. Anterior cavity is broken up into anterior chamber, which is in front of your iris, the colored part of your eye, and then there is the posterior chamber, which is just behind the iris, but in front of the lens. Okay, this is the lens, the optical focusing, the primary optical focusing component of the eye. All right, so this anterior cavity, both posterior chamber and anterior chamber, are filled with aqueous humor, and then the posterior cavity is filled with vitreous humor. We'll talk about the distinction between those in a little bit as well. We have, as an extension of this uvea and choroid, we have the ciliary body here. The ciliary body is responsible for helping adjust the shape of the lens. All right, your focusing power, your accommodation. We'll talk about that in much more detail as we go on. So light comes in, goes along this visual axis, and hits this point here. This point is called the fovea. The fovea is at the center of what's called the macula, which is this whole region right around here. Macula just means disc, like a large spot. And then the fovea is a divot that is in the back of the eye, the fovea. This is the center of your field of view. It's the center of your focus. When you're looking at something, the light from that thing is being reflected directly onto the fovea. Then there is the optic disc. This optic disc is where the optic nerve enters the eye. This is not your center of focus. This optic disc actually represents a blind spot. We'll see how that works in a bit. So that's all the important bits that you need for today on that. All right, the three layers of the eye. There is this fibrous layer, as I initially said, composed of both the cornea, the clear part over the anterior cavity, and the sclera, the white fibrous part that makes up the body of the eyeball. The sclera, the job of the sclera is to support the eyeball. It is to protect the eyeball. In fact, it is also important in terms of regulating, in terms of helping to adjust the shape of the lens. And we'll understand that in a little bit when I talk about the ciliary body and how it interacts with the sclera. Also, the cornea does add some small amount of focusing power to the light. Has anyone here had RK or LASIK, radial keratotomy? You guys aren't quite old enough for it yet. But any of you who wear glasses or contacts, you can get the cornea etched with lasers that will, it's like shaping that to add a contact lens, the focusing power, change the focusing power of the cornea. The second layer is called the vascular layer. It's the uvea. And it's made up of three primary components. First is the choroid, right here. This whole region along here is the choroid. Choroid, you've heard this term before. Where have I used the word choroid? What's that? Yeah, the choroid plexus. And what did the choroid plexus do? What was its job? Where was the choroid plexus? You guys remember? It was inside, was it in the kidneys? Was it in the kidneys? No, it wasn't in the kidneys. I haven't talked about the kidneys yet. What have I talked about? I haven't talked about too many organs yet. I've talked about
plexus, right? That's neurological.
It's in the brain. Yeah, it's in the brain. It is tissue that is made up of ependymal cells that line the ventricles of the brain and is responsible for making cerebrospinal fluid. The choroid plexus makes the CSF. All right? Yeah. So the choroid here, its job is to make the aqueous and vitreous humor, very similar to the choroid plexus in the brain. Okay? Aqueous humor is not CSF. It's not, vitreous humor is not CSF, but it is the vascular layer. It's fully invested with vasculature. It then blends into the ciliary body, which is a muscle component. It's made up of the ciliary muscle, the ciliary process, and then the suspensory ligaments. We'll see how that works in a bit. And then finally, there is the iris, and the iris has pupillary dilator and constrictor muscles in it that are responsible for changing the amount of light that gets into the eye. It's like, is anyone here into photography? With an SLR camera, you're changing the aperture. That's the iris. And then finally is the retina, and this is the neural layer of the eye. This is where all the photoreceptors are. This is where the magic happens. We'll see how that works in hopefully exquisite detail, assuming that I can keep my lecture moving here. So the pupillary muscles. Here are the muscles that lay beneath the iris, within the body of the iris. So you have the dilators, which are radially disposed around the pupil. They contract, pulling that aperture wider, allowing more light into the eye. We have pupillary constrictor muscles that are circumferential, and when they contract, they close that aperture off, the sphincter, the pupil. All right. So when there is high light, when there's a lot of light, pupillary constrictors go. When there's very low light, pupillary dilators. You may ask yourself in this photo, how come this guy's eye is fully dilated when you see these bright lights shining, reflecting in his lights? Well, they put a chemical in his eye that interfered with the autonomic innervation of the activity of the pupillary dilator muscles and made them big. The compound was it's derived from nightshade. I think that's right. Yeah, it's nightshade, right? Which was the derivative of which was used by women in the Victorian era. They would put drops in their eyes because it made their pupils get really big, and they thought it was beautiful looking to have these large, dark pupils that people could look deeply into, or whatever. But then they'd stumble around because they couldn't see properly. All right. How many here have had their eyes checked before? Probably a lot of you. Yeah, great. So when you do that, used to be who's had the pressure in their eye checked before? Okay. How did they do that to you?
They put some kind of anesthetic on it, and then they just sort of poked it with this little cone-shaped device.
Old school, yeah. That's the old school way of doing it. Has anybody had it where they just puff the air onto the eye? Little puffs, yeah. That's more common nowadays. It's not the only way, but the way that Jacob's describing here is how it used to be done for many years, where they would actually put a sensor right onto the surface of the eye and determine the pressure in the eye. So what are they actually measuring? They're measuring the fluid pressure of the aqueous humor that's here in the anterior cavity. So here's the posterior chamber, the anterior chamber, all of it's the anterior cavity. The aqueous humor, this fluid, is getting produced by the choroid here. It's getting pumped out into this anterior cavity, passes through the pupil and into this anterior cavity, and is resorbed by the structure, the analog of the arachnoid granula in the brain, right? The arachnoid granulations were how we resorbed CSF, and in the eye, it's through this sclerovenous sinus or the canal of Schlemm. There's about 3 or 4 different names you can find out there for this structure, but it lays in what's called the limbus. So if you look deeply at somebody's eye, you'll notice this point where the iris meets the sclera, the white of their eye, where it looks a little bit milky. It's this sort of milky ring around the edge of the colored part. And we call that the limbus of the eye. Limbic meaning liminal. It's related to the word liminal, meaning sort of on the margins. But beneath the limbus is this sclerovenous sinus, and that's how we resorb the fluid into the venous circulation. So, and they're able to take the pressure. The problem with intraocular pressure, it helps retain the eye shape. What happens, do you guys know the name of the disease if you have intraocular pressure that goes too high? What is that called? Say it. Glaucoma. Yeah, glaucoma. Does anyone have a relative that has glaucoma? My mother does. Yeah, glaucoma is a problem when it's like hypertension for the eye, kind of, right? That intraocular pressure is too high. And the problem is when that intraocular pressure goes higher than the arterial supply, blood supply pressure to the eye, then blood can't get to the eye, and the retinal photoreceptor cells begin to die, and so the person goes blind. Having high intraocular pressure is a good way to die. Is a good way to go blind. Okay. All right. So here is a picture of the vitreous body resected from the eye. That's pretty crazy looking picture there. The vitreous body is actually a solid gelatinous mass. And despite that fact, it is still 99% water. There's some sugar, salts, sugars, this protein called vitricine, which helps the optical clarity of the vitreous remain intact. And then we have some collagen and high hyaluronic acid that is also in there. And the high hyaluronic acid is what is responsible for maintaining such a high percentage of water. High hyaluronic acid is very hard to dehydrate. Okay, the lens itself is made up of these fiber cells who, as they develop, here are the margins, the nascent fiber cells, they have, you can see organelles, nuclei, etc. But as they mature, those organelles and nuclei are broken down and resorbed, and instead the cells just pack themselves full of these proteins called crystallins, much like vitricine, that helps to fine-tune the optical properties, the optical clarity, and focusing power, the optical refractivity of the lens. Okay, so that's what the lens looks like. Here's an EM of it. You can see the lens is attached to the ciliary body back here with these suspensory ligaments, the little ropes that help secure it in place. Yeah.
Those are meiotic for their entire life.
What's that?
Are those meiotic for their entire life?
Yeah, they're non-replicating, right? So once they have gotten rid of the nucleus and all the organelles, they're quiescent. Okay, so one of the things you'll notice here is someone with a slit lamp taking a picture of this guy, and which eye do you think this person is looking at you with? This one or that one? Who says what is that to you guys? The left. Left, yeah. It's red because you're able to see through the lens to the retina. You're looking at the retina. It's red because of the retina. This one is milky because he has a cataract. He's probably had the cataract removed on the other side, in fact. The cataract happens because chronic exposure, he doesn't look like a spring chicken, chronic exposure to UV light leads to cross-linking of the crystalline proteins that are in these fiber cells of the lens, and that reduces their optical clarity. Okay, and then with time they form a cataract. A cataract is a waterfall, and that's what it was. I guess the word is originally comes from the Greek, but it means the waterfall, that the curtain that comes across your vision. So, why did I put this one up here? I don't know. More anatomy. Maybe I'm not sure if there's a point that I need to be making here, except that I suppose I already made this point. The light comes through the visual axis and lands on the fovea at the back of the eye. I'm not sure. Maybe I'll go back to it if I need that picture again. So, refraction definition: bending of light by any by the passage of light from one medium into another medium of a different optical density, right? So, and that describes air versus the medium of your lens. So light is bent. Here comes light from a distant source. It hits the lens, and it gets bent. This point from the center of the lens to where all those rays of light converge is called the focal distance. All right, and that focal distance changes depending upon the relative disposition of the thing that you're trying to bring in focus. Okay. So you move the source of light further or nearer, and you're going to affect that focal point. All right. So, no. What am I talking about? I'm talking about inversion, right? Got myself lost here for a moment. So the way it works in the eye is when light comes in, it passes through the lens and gets thrown onto the back of the eyeball. When you're sitting here and I'm looking at you, I see the clock here. That image, the light that's reflecting off that clock, is being thrown down onto the inferior margin of the retina, on the bottom of the retina. As I look at Logan's hat, the light from that clock is on the bottom of my retina, and the light from my computer screen down here is on the superior margin of the retina. So there's this inversion. And it's just the labeled line code. All those neurons that are taking the signals from that get labeled, sent back to the brain, and our brain interprets it so that I know which way is up, right? I don't see the world upside down. Am I making it worse? It's the same side to side. It's the same side to side. So light from things on the left is thrown to the right of the retina, and lights from the left is thrown onto the right in the back of the eye. Is anyone confused by that? I feel like yes, no? Okay. Here's a good one to know: accommodation. So accommodation is the process by which a lens changes its shape to change the focus on the retina. All right. So, here is a rounded lens. The ciliary muscles are contracted. There's a mystery here that I want you to solve. Ciliary muscles are contracted. The lens is round. Okay, and it's a near thing. So we've rounded the lens and thrown that point of focus onto the back of the eye. Now we're looking at something. We're going to remove this point far away, so these lines are much more parallel. We need a less rounded, less focusing power in the lens. The ciliary muscles relax. The muscles relax, and the lens goes from round to more flattened to keep that point of focus on the back of the eye. How is this happening? Why is it that the muscles are contracted and have a round lens, relieving pressure. I'm sorry. They're contracted. They're not pushing on those. They're not pushing on those ligaments. How is this rounding when those muscles are contracting? Does anyone have any theories as to what's actually happening here? What the dynamic is? Yeah.
Are they attached higher up? So there's kind of like they're pulling this way to squish the thing like that. Does that make sense?
They are. I'm not sure. I'm going to interpret what you said the way I understand what you said to give you the correct answer, which I'm hoping is what you intended. The sclera is almost like a spring force this way. The muscles contract, and as the sclera has this spring force, it's pulling on the sclera. The white part of the eye is like this. It's pulling on these ligaments. It's pulling on these ligaments, flattening the lens. All right? Flattening the lens. Those muscles contract and slightly bear down on that spring force from the sclera and allow the lens to relax into a more rounded shape. You guys see how that works? So they're not pulling directly on these ligaments at all, in fact. This is a very crude drawing. What they're doing is relieving a little bit of the circumferential spring force of the sclera that here, when they're relaxed, is tugging on these ligaments and flattening that lens out. They contract like this, relieving some of the spring force from the sclera and allowing that lens to relax into a wider, rounder shape. You follow me? Does that make sense a little bit? It's pretty ingenious.
What direction are the muscles aligned?
I think they're circumferential.
Okay. So then, how do they get thicker? So do they?
The lens? The lens is what's getting thick. It's allowing to become from flat into a rounder shape.
Yeah, the lens gets thicker, but then if it's a circumferential muscle that's contracting, does that? Oh, now I get it, because it's actually bringing the base where the ciliary bodies are closer together.
Absolutely.
Okay.
Astigmatism. Anyone here astigmatic? Yeah. Anyone severely astigmatic? Take your glasses off. A person who is not astigmatic sitting out there looking at this diagram is going to see these bars being equidistant from one another, the same arc distant around that circle. Someone who's astigmatic will not see that, however. There is an abnormality in the shape of their cornea, all right? And it's a problem. People with severe astigmatism have a hard time wearing contact lenses. You can't get a contact lens to fit a person with astigmatism very well if they're severely astigmatic. The problem is you have multiple focal points on the back of the eye, and it can give these people very bad headaches. So they have to have corrective lenses that account for this astigmatism. All right, visual acuity, here's some more definitions. Myopia, or short-sightedness. A person who is myopia has their focal point in front of the retina, so we get a diverging lens in front of the eye and throws the focal point back onto the retina. Emmetropia is 20/20 vision. It's normal vision, supposedly normal vision. Hyperopia is in contrast to myopia. Hyperopia, or farsightedness. This is a person whose focal point is behind the retina. All right? So we get a converging lens, and we bring the focal point back up there. So elderly people often with time become hyperopic as the lens itself is not as elastic and is not able to go back and forth. We have this cross-linking that we talked about, and the lens is not able to regenerate itself in the way that other cells are. So we call this presbyopia, like a presbyter in a Presbyterian church. A presbyter is an elder. So a presbyopic person is a person with the hyperopia of the aged. Some pretty standard definitions there, straightforward. Okay, so let's look at the retina. We have the fovea, the little divot, right, where it's the center of our field of view. It's located in the macula. We have the blind spot. This is where the optic nerve attaches. We have the ophthalmic artery and the retinal artery and veins. So this is what's called a fundoscope here. Somebody's taking a picture of the fundus of the eye, the retina, the back of the eye. When you have a neuro-ophthalmologist come in to see you, they, at least the old school ones, will have a red pin in their lapel, and they'll pull that out because you can map your own blind spot. It's about 23 degrees off your visual axis to the lateral, 23 degrees lateral from the visual axis. So if you're looking at a point, you can take this red pin and get it just right where the head of the red pin will disappear and you'll no longer be able to see it. Your brain just takes everything around it and sort of smudges it in because it's in your periphery. It's not that big a deal. It would be the light falling directly onto that blind spot. You can demonstrate that here with this by looking at the X with, so how would this work? You would have to look at the X with your right eye, cover your left eye, and probably won't work for quite everybody in the class, but for some of you who are in the right spot, you should be able to get it so that that dot disappears. All right, and where that light is falling right on top of the blind spot. The point being that a neuro-ophthalmologist is looking at, they want to know what's up with the retina, and is there a problem with that disc, and is it getting bigger? Is your blind spot or scotoma, the word is scotoma for a blind spot, is it getting bigger? So there's a lot. What's amazing about the optic nerve, and I don't want to get too sidetracked here, is that it is the only nerve in the body. It is the only nerve in the body that can be visualized non-invasively. You can look at the optic nerve. Can't do that with any other nerve. It reports on a whole lot of physiology. A good neuro-ophthalmologist who knows what they're looking at can diagnose a vast array of things in the body, not just related to the eye. You can see if a person has multiple sclerosis, neuromyelitis optica, various infective processes. Is there a problem in the cranial arteries? Is the person on drugs? Are there brain tumors in that person's brain, increasing the intracranial pressure, pushing on the back of the eye, leading to cupping in the optic cup? Have they had radiation? So a lot of these are being depicted here in these various fundoscopes. A neuro-ophthalmologist who knows what they're looking at when they look at an optic disc can tell a tremendous amount of about your state of being. When you have somebody look in your eye, or if you are getting trained to look into somebody's eye in your training, it's not the time to be shy, right? When you're looking in somebody's eye, you have to take an ophthalmoscope and get right up on that person, so you can actually see into the back of the eye. This is how it's done. This woman looks sadly unaware that this guy doesn't know what the hell he's doing at all right? Because he can't see anything in the back of his eye from back there, in the back of her eye from back there. So when you're looking at the fundus, get a good look. You get in there and you get a good look. You'll all have the chance if you go to medical school. They will make you do this. Don't be shy. Get in there. Make sure the person is teaching you the right way. So now the way that is visualized in a modern neuro-ophthalmology office is with this optical coherence tomography, this high-def optical coherence tomography, OCT. This is actually a picture of the fundus of my eye. This is the fovea on the right and left eye. And there's a whole host of data here that I actually am not going to take the time to walk you through. But that's what it looks like nowadays. They can get extremely high resolution computed tomography images of the back of the eye. All right, so kind of took more time on optics than I meant to because this is the fun bit. Photoreception, rods and cones. Rods and cones. There are two visual systems. There is scotopic vision and photic vision, or color, right? Scotopic vision is light and darkness, all right? This is done by the rods. It's highly sensitive. A single photon can trip one of these rods. A single photon will trip a rod and lead to photosensation. This helps you. This is night vision, right? Cones are less sensitive, but they're very highly resolved. They're densely packed. They give you high resolution imaging. Scotopic vision is not high resolution. It's not high resolution. This is a trade-off that we have. High sensitivity versus high resolution. All right? Very low light will set these off. These require more light, but they're densely packed and give you high resolution vision. They're densely packed in the fovea. We find cones in the fovea. Leads to color vision. All right? The way the retina is organized, light comes in. The photoreceptors are the deepest layer. The light has to pass through several other layers before it even gets to the photoreceptors. It passes through these ganglion cells, then a layer of bipolar cells, before it gets to the photoreceptors. At these junctions between the photoreceptors and bipolar cells, we have these horizontal cells, which are intervening cells, and then also at the layer of ganglion to bipolar, there are these amacrine cells, which are intervening cells. The thing that should be immediately apparent to you is that there is an enormous amount of neural processing of visual information before the signal even leaves the retina. Before the neural signal even leaves the retina, there's a huge amount of neural circuitry and information processing that's happening. All right? Before it even gets to the optic nerve, a whole lot of information is getting sorted out right here. And it's far beyond the scope of this class to get into the depth of that, but we'll see a little bit more. So let's see what it looks like. Let's look at one of these. Rods have these invaginations of the plasma membrane that form these sort of discs, and there are genuine lamellar discs in the cones. The heads of both of which are embedded into this pigmented epithelium because if any light gets past any of these photoreceptors, we want it to get picked up by the melanocytes in the back of the eye so that it doesn't damage any deeper tissue, right? We got to stop the light at that point. These melanocytes are also responsible for recycling bits of the exocytosed older discs in some of these cones here. So in looking at one of these stacks in the photoreceptors, we see that there are these lipid bilayers in these stacks with this protein that we're going to call rhodopsin embedded in it. All right? Rhodopsin is the conjugation or the combination of a protein called opsin and this chromophore, this light-absorbing chemical called retinal. retinal. In fact, when it's ready to go, it's 11 cis retinal, referring to this double bond here. We'll see what that means in a bit. All right, retinal, vitamin A. Comes from vitamin A. Okay, so photoreception. This is the money here. It's incredible how this works, and I do not have a very good explanation for why it works this way. I can tell you how it works. It's an active area of debate. I can hypothesize. I cannot give you an answer. I don't think anybody can. But this is how it works. All right. Normally, in the dark, when a photoreceptor is not getting stimulated, there is this dark current. Remember, this is a neuron. This is a neuron. Here, down here in the body, there's the sodium-potassium ATPase that's pumping sodium out to beat the band, right? Pumping sodium out. But there's channels up here near the photoreceptors that are open that are allowing the sodium to come in. It's just coming in. It's called a dark current. It is depolarizing this cell. In the darkness, the cell is depolarizing and signaling neurotransmitter release onto these bipolar cells. Okay? Maybe the opposite of what you would have expected. How does this work? Well, there is cyclo-GMP. This is a G protein coupled receptor here. Or a channel, pardon me, it's a gated channel. So the channel is open, sodium is flooding in. Rhodopsin and these accessory proteins are inactive. So a photon comes. It transmits through the cell and hits the chromophore in the rhodopsin, the 11 cis retinal. Here it is, retinal, this long chain of conjugated double bonds. This is highly conjugated aromatic compound. All right? 11 cis. This is a high energy structure. This is a high energy structure. A lot of pi electrons for those who's had organic chemistry so far. A few of you know what pi electrons are then. These pi electrons in this double bond are open to the absorption of light energy, and they do. And it's just enough of a bump to cause an isomerization, where 11 cis isomerizes to the lower energy all trans retinal. Okay? Molecular switch. When that happens, we're going to start a cascade of events. 11 cis retinal becomes trans. That's going to signal to this protein called transducin, which then signals to phosphodiesterase, which is then going to hydrolyze cyclic GMP to just GMP. And cyclic GMP is going to diffuse away from this G protein embedded in this G protein channel here, sodium channel. The door is going to swing shut. Sodium is not going to be able to influx anymore. And we are going to interrupt the dark current, stop neurotransmitter release, because we're going to return to the resting potential, which should be the resting potential of the membrane.
Sorry, can you go back a slide? I missed what's happening with the GMP.
CGMP gets hydrolyzed by the phosphodiesterase, the PDE, and CGMP begins to diffuse off of these cells, or these receptors, these channels. Door swings shut. Sodium is no longer allowed in.
Okay, so the phosphodiesterase is attached. That's triggered by the formation of trans retinal.
Yep. Yep. So When this inhibitory neurotransmitter release stops, this bipolar cell says, time to go. And it does. Okay? That is like the kindergarten version of what's actually happening here. This here is a little bit more detailed depiction of this synapse. It is a wildly complex synapse that has all these horizontal cells coming in, mediating the types of interactions that are happening. This is perhaps the most complicated synapse found in the body. All right. I'm not going to go into what this all means in depth. I couldn't, frankly, off the top of my head. It's quite complex. But yeah.
Sorry, so the light passes the bipolar cell and hits the cone.
Yeah.
And then the cone affects the bipolar cell in front of it?
Yep.
Okay.
Absolutely. All right. So after this happens, we have rhodopsin, retinal, 11 cis retinal absorbs a photon, becomes 11 trans, and then we get bleaching. When this happens, it sets off the cascade with transducin and PDE and CGMP and all that business I just talked about. But it also, 11 trans, or all trans retinal, dissociates from opsin. So opsin's here, and we use some ATP, we burn some energy to re-isomerize this to the high energy 11 cis, which then reassociates, reconjugates with opsin to regenerate rhodopsin. Okay, that's the photic cycle, recovery after photic stimulation. So bleaching, as I stand here talking to you, I am often accosted by this bulb up here, right? And it's super bright. If I am unable to resist temptation and I look at it, and then I look back at you and I look at Sonia, suddenly her face is this bleached out blob that I can't see because I have photo bleached the retinal off the opsin. Okay, like all of the retinal has been bleached off of all of the opsin on those rods and cones in my eye. And it takes a while for that to build back up. All right, we're getting close here. Let's see, maybe one more slide. So scotopic vision, dark vision. This is the vision conferred by the cones. Night blindness, however, is where you do not have enough retinal to conjugate to the opsin in the cones. Was that?
Scotopic is cones or rods?
Scotopic is rods. What did I say? Did I say cones? Pardon me. Yeah, it's the rods. Thank you. Here's vitamin A, retinol. It gets converted to retinal by getting oxidized here. Beta carotene is found in carrots and other vegetables. It's a lot of bang for your buck. A single molecule of beta carotene, once oxidized, the double bond gets cleaved and oxidized. We get two retinals out of a single beta carotene molecule. So a person who is night blind, transiently night blind, has a vitamin A deficiency. All right, I wanted to leave it there. There's like another slide or two that I'll finish off on Wednesday. My ear lecture is much shorter, or a little bit shorter, so I'll be able to finish up on Wednesday with the dual vision system. I'm going to talk about scotopic versus photopic just a little bit more. Are there any questions? Yeah.
So you're going to talk about bipolar cells later?
A little bit,
Okay.
a little bit. Yeah, not in any depth.
Okay,
though.
but just that signaling, how does that affect the bipolar cell?
Yeah, bipolar cell then, so I will talk about it a little bit here. Bipolar cells are going to signal onto a ganglion cell, which then,
Okay.
sends the downstream signal. We'll get to that.
Here's what neurotransmitter are the cones and rods releasing?
GABA, I believe.
Okay, so they're that makes sense. They're
GABAergic, yeah.
it and then turn off.